And today we are super lucky to have Yucheng Wang that will present the last Nature work that was published a few weeks ago. In this work, Yucheng, Mikkel Pedersen, and Inger Alsos are first authored, and there are many other authors there, including Alex that is here today. You will talk about reconstructing 50,000 years Arctic environmental history using ancient eDNA. And Yucheng is now sitting in Cambridge, the University of Cambridge sorry, I have to make that, sorry. Okay, in the University of Cambridge in UK. And I don't know exactly where you will go later. So if you can explain a bit where you are and where you want to go, that would be nice. So thanks. Your screen is shared, so I think everything is good.
Okay. Am I free to go?
Yes.
Yep. Okay. Great. Thanks, Enrique. And hello, everyone. Good afternoon. My name is Yucheng Wang, and I'm a postdoc at Zoology Department and Cambridge University. And my topic today is reconstructing 50,000 years Arctic environmental history using ancient eDNA. So the idea, as Enrique mentioned, is basically to cover the new findings and the new method development involved in this recently published Nature paper. Oops. So the motivations for carrying out this project. So despite there are many studies using different approaches to reconstruct the flora and fauna successional history in Arctic in late Pleistocene and Holocene, there are still a lot of questions remaining highly debatable. And we realized that in order to really, directly address these questions, the best option would be to apply a uniform method for paleoecosystem reconstruction across the whole Arctic, covering this 50,000 years time period. So we collected more than 500 sediment samples from 74 sites across the Arctic. And by shotgun sequencing the ancient environment DNA preserved in the samples, we generated this eDNA data set, which contains more than 10 billion quality controlled eDNA reads. And we also sequenced more than 1,500 modern Arctic plants to be used as reference. And by attaching these Arctic plant genomes into the public, NCBI databases, we eventually generated a very comprehensive reference database, which contained more than 308 million entries covering more than 1 million different organisms. And by mapping our eDNA rates against this huge reference database, we successfully reconstructed the flora and fauna communities in the last 50,000 years. So first, we put all the reconstructed vegetation into one picture to looking into kind of the general Arctic vegetation formation and the Arctic vegetation as a whole, how it changed in responding to the climate change. So we found first is that before and during the LGM, the last glacial maximum, there was this extensive steppe tundra, a very homogeneous vegetation dominated across the Arctic. And this vegetation, homogeneous vegetation, contained both steppe and tundra element, but without trees. So an extensive open steppe tundra distributed across the Arctic in the Pleistocene. And if we're looking into the diversity change, we found that vegetation diversity was very, very sensitive to climate change. So if we're looking into this pink curve here, we see that the diversity changed dramatically within this 50,000 years time period. However, the trend is not always following the same way. But for example, before the LGM, when the temperature decreasing, we saw that the vegetation diversity increased in response. Well, when the time entering the LGM, the extreme cold and dry condition occurred, the overall diversity, vegetation diversity declined dramatically until the Younger Dryas when the Pleistocene Holocene transition happened. And the vegetation then thereafter increased again as a response to the warming climate in the Holocene. So it's very sensitive to climate, but in different ways. And if we're looking into the morphological form of the vegetation, what we found is that when the woody proportion increase, we have the overall vegetation diversity decrease, where when the herbaceous plant increase, we have the overall diversity increase. This indicates that abundant multiple different herbaceous plants turn to share, co-appear, and share the landscape. Well, when the environment gets more suitable for the woody plant, a few of these species would more likely to dominate the ecosystem. And within the herbage, forbs were more abundant than grasses, but not as dominant as previously suggested by the metabolic coding study. And next, we divide the vegetation into 4 regions to kind of looking into the regional patterns. And what we found very obvious is that the region which used to be covered by this huge ash sheet has experienced most dramatic vegetation changes, both in terms of the diversity, also in terms of formation. For example, for North Atlantic, which is basically Svalbard, Greenland, and Iceland. So the vegetation changed very much. Well, for the region where was very much continental, like northwest and central Siberia, the vegetation maintained basically unchanged. So the conclusion here is that continental ice sheet is the key in driving vegetation change in the Holocene. So for the megafauna distribution, we combined our eDNA identified megafauna data site with a huge fossil megafauna database to reconstruct the distribution map of different megafauna species in the Arctic. What we found first is that basically all the species of megafauna, their distribution range continuously shrink along the timeline. And we found many late survival of megafauna species. For example, for woolly mammoths, which used to be got extincted on the continental Arctic around 10,000 years, but we found they still survive on the Taimyr Peninsula of Central Siberia until 4,000 years ago. And we also found woolly rhinos to Holocene and horse in North American to around 8,000 years ago, and bison in Siberia into even mid-Holocene. This late survival of megafauna is kind of expected because when a species approaching to extinction, its population size must be very small. And an individual species only left one skeleton after its death, while it spread its DNA, millions, billions of DNA sequences into a wide range of landscape every day throughout its lifetime. So it's much, much more likely to find the late survivals by eDNA compared to the traditional, fossil-based chronology method. So this late survival of megafauna also suggests that this species coexisted with human for at least a 20,000 years interval, which disagrees with the human overkill theory for causing the megafauna extinction, which assumes the extinction happens in a few centuries after the first human contact. And we also developed a method to use eDNA metagenomics directly place the identified species into a phylogenetic tree, onto the phylogenetic tree to haplogrouping the identified taxa, K taxa. And here is an example for the mammoths. So first, it's the eDNA identified haplogroup are very consistent with the fossil remains. So if we're looking into this clade distribution map, we found that before the LGM, there are all these 4 different mitochondrial clades well distributed across the Arctic. Well, after after the LGM, only two clades survived, and when it comes to Metallocene, only the final clade survived. And we found that this late survivals detected by eDNA on Taimyr Peninsula belongs to the same mitochondrial clade to the late survivals on the Wrangel Island, which is a small island, well known for some mammoths survived in 4,000 years by fossil, of course. However, if we zoom into the phylogenetic tree to looking into the detailed patterns, there's none of these Taimyr samples placed into this small, Wrangel clade here on the top right. These two clues combined suggest that this two late survival population has some sort of a genetic relation to each other, but they are not belong to a same population. And we also found some previously unsampled clades. For example, if we're looking into this top sample, is CR511 here, there are 27 SNPs, mitochondrial SNPs available for this sample, but none of them support it to be placed into any of the 4 known clades. And the best placement for this sample was the shared root of clade 2 and clade 3, which means we must have some, woolly mammoth population we haven't sampled yet. So we can't really place the sample into a fire scale. So the driver for the megafauna extinction, this is I guess this is for most of the people, it's the most interesting topic. So if we put the mammoth distribution map along with the vegetation, regional vegetation changes, what we found is that the survival of this megafaunas, in this case the mammoths, is very much related to this previous mentioned, this open steppe tundra vegetation dominates the Arctic during the Pleistocene. So for where we have the late survivals, for example, for mammoths here, we have it in Central Siberia until 4,000 years. The vegetation in this region in Central Siberia also maintained pretty much unchanged. So the vegetation change seems to be the reason for mammoth extinction. And next, we developed this model to comprehensively put different components of climates, human distribution, vegetation, and animals into one model to kind of predict what is how different elements contributing to the megafauna distribution. And what we found first is the herbivorous animal tend to co-occur in timeless space. For example, for here, the best predict variable is actually for hare's presence is the mammoth's presence and caribou and the horse and bull presence. So it's obvious to us that this Arctic megafaunas applied a coexistence strategy instead of interspecies competition under this extreme Arctic climate condition, which makes perfectly ecological sense. And we also found that human was basically unrelated to animal distribution. If we're looking into the bottom lens of the figure B, which is the human contribution, we found human was basically neutral for all animals, only except for hare. And if we're looking into the plant, we found that the plant NMDS, the plant component 1 and plant component 3 generally had a negative effect on the megafauna distribution. Well, the component 1 basically represents an aquatic against terrestrial herbaceous plants, while the component 3 generally represents a woody against the herbaceous plants. So the increase of wetland plants and woody plants, the forest, had a negative effect on the megafauna distribution. And if we merge all the herbivores together into one factor, which makes sense because they are highly likely to co-occur, right? And we found temperature is actually not significant. Well, it's the participation mainly is the pre-simple negative factor in driving the megafauna distribution. So in short, human was not significant, and the increase of participation and the increase of the wetland plants and the forest had a negative effect and was driving the extinction of this Arctic megafauna species. And along with this analysis, we also developed many new methods for eDNA metagenomics, which include 2 new eDNA extraction protocols and a new pipeline for assembling plant genomes. And we also applied the DNA damage degree to authenticate taxa identification from ancient eDNA. And we developed a KEMA-based method to evaluate the reference database availability. And using this method, we found that the reference is extremely important in eDNA metagenomics-based taxa identification. And we also developed a new method to quantify the taxa based on a few of variables, including DNA damage degree, sequencing depths among different samples, reference database availability for different taxa, and also the taxa's genomic distinctiveness. And we also did a large-scale comparison of Asian eDNA metabarcoding against the metagenomics. And also this method for haplogrouping the eDNA metagenomic data identified species to achieve phylogeny, as I shown before for the Mammoth case. And this last method, this model to comprehensively incorporate different components into one model to predict the effects of different factors on the animal distribution. So the takeaway message, for the vegetation, we had this very homogeneous steppe tundra flora dominate the Arctic before and during the LGM, followed by regional divergence of vegetation in the Holocene. And many megafauna species survived much longer than previously known. And the ice sheet is a key for the vegetation change during the Holocene climate warming. And this persistence of the steppe tundra vegetation is key to enable the late survival of several megafauna species. Human is not significant in driving animal distribution, where the higher participation and increase of the wetland plants are the main negative effect on the animal diversity. And phylogenetic analysis of eDNA metagenomics identified species is possible and makes perfectly ecological and genetic sense. So we have many new methods developed, and please go check the paper if you are interested in this. And that's basically it. Thanks for all the co-authors and collaborators and funding bodies to make this happen. And thanks you very much for organizing this seminar and for your attention. Thanks.
Thank you very much, Yucheng. So cool data set. The first week that it was released, we were already free in the same office to try to download it. So I'm sure many people are reading this paper and trying to see all what you did. So we will have questions from the people listening today. So feel free to just say who want to start the question. You can write on the chat also, and I can read them. So who would like to start? Vivian? No. Yes, you said yes. Vivian, you can start if you want.
Okay, great. Hi. Thanks so much. That was really interesting. I had a question about the modeling of the human impact. So if I understood correctly, it's not based on human DNA that you find in the samples, right? It's somehow other factors. Can you explain that a bit?
So basically it's niche modeling of the human distribution, right? So because we want to be conservative for this model, right? So so it's actually not really human distribution. It's rather the all the suitable environments which can potentially support human back to the whole time period. So we purposely extended the human distribution in this case in order to test if they have an effect on animal distribution, right? So it's niche modeling of the human suitable environment in the Arctic.
Okay.
Does that answer your question? Well, it's a completely another field. I mean, I learned a lot by myself as well.
Yeah, I guess I would have to dig a bit more into it, but so you're essentially saying you check what environment in terms of temperature and in terms of sort of...
Yeah, so basically the idea is that, so we have the human, like the fossils and all the archaeological sites in the Arctic, right? And they built up a model to evaluate, the climate conditions, environmental conditions in these sites, and then simulate across the whole Arctic to find the same site which can potentially support human. And we identify all these sites as human distribution. So extended is it greatly is the human distribution in this case.
I see. Okay, yeah, that's clear. Thank you.
Yeah.
Nikolai, you can ask a question.
Yes, hello. Hi, Yucheng. Very nice talk. Thank you. I was just wondering, the woolly mammoth that you found on Taimyr 3.9 thousand years ago. So what was the number of reads? What was the quality metrics this detection was based on? Like the number of reads, breadth of coverage. Like, could you please let me know?
Yeah. So it's not a single sample, right? It's a series of sample, continuous series of sample, and in multiple sites. So it's not a single sample supporting the latent survival. It's like 30 different samples from 10 multiple sites support the same series. And secondly, for the threshold, first we have of course the reads number, and then it's a series of authentication methods and also the damage patterns. And even as I mentioned before, to be conservative, we even, so for example, for woolly mammoth DNA, right? If we find a DNA uniquely, exclusively mapped onto a mammoth genome, we then give 2 mismatches to this reads against all other species in the reference database. All this, 1.4 million organisms, the genome for all these genomes, with 2 mismatches. If they mapped onto other species, we remove these reads. So that means each of the reads we are talking about here, first is uniquely mapped onto woolly mammoths, and secondly, even with 2 mismatches, it mapped to nothing else. Is that make sense to you?
Yes.
Answer your question? Yeah.
Nice.
Cool. So I have a question by myself, but obviously I'm a microbial ecologist, so I have not relevant question, not smart enough. So I will ask a technical question. What kind of setup do you think you would have done differently if you could? Do you think you miss sampling site or things that would have been super useful to answer better the question you try to answer?
Yeah, definitely. We know we missed a lot of the information, right? For example, we have many other species like, lion, like, liming, all these small like animals, and also, camel or even, right, Arctic camel. And simply because we don't have a good reference for them, so we can't confidently and reliably identify them. So we don't include them in the results. So we know we definitely missed something.
you think sequencing depth could have been also a way to catch
Well,
them?
I think mainly for this species, it's mainly about the reference genomes. So we don't have reference genomes. So we, based on the current methodology, we can't really do anything, right? So without a good reference or without even available reference for them.
So you have to go back and get some Arctic camel's bones.
Yeah, that's the ideal.
That's a new field trip you can plan for the next years. Super cool.
Yep, thanks.
Okay, we have other stuff to discuss in this seminar, but if you have a few more questions, you can ask. We can stay 30 seconds more. I will close the recording so I can stop the video here.